DNA gets a lot of attention as the store of our genetic information, and it deserves that. If we didn't have DNA, there would be no way of keeping the information that makes us us and other organisms what those organisms are. And DNA has some neat properties. It can replicate itself, and we go into a lot of depth on that in other videos. So DNA producing more DNA, we call that replication. But just being able to replicate yourself on its own isn't enough to actually produce an organism. And to produce an organism, you somehow have to take that information in a DNA and then produce things like structural molecules, enzymes, transport molecules, signaling molecules that actually do the work of the organism. And that process, the first step, and this is all a review that we've seen in other videos, the first step is to go from DNA to RNA, and in particular messenger RNA. And this process right over here, this is called transcription. We go into a lot of detail on this in other videos. And then you want to go from that messenger RNA, it goes to the ribosomes, and then tRNA goes and grabs amino acids and they form actual proteins. So you go from messenger RNA, and then in conjunction, so this is in conjunction with tRNA and amino acids. So let me say plus tRNA and amino acids. And I'll write amino acids I'll write it in a brighter color since that's going to be the focus of this video. So tRNA and amino acids, you are able to construct proteins. You are able to construct proteins which are made up of chains of amino acids. And it's the proteins that do a lot of the work of the organisms. Proteins, which are nothing but chains of amino acids, or they're made up of sometimes multiple chains of amino acids. So you can imagine, that's an amino acid. That's another amino acid. This is an amino acid. This is an amino acid. You could keep going. So these chains of amino acids, based on how these different, based on the properties of these different amino acids and how the protein takes shape and how it might interact with its surrounding, these proteins can serve all sorts of different functions. Anything from part of your immune system, antibodies. They can serve as enzymes. They can serve as signaling hormones like insulin. They're involved in muscle contraction, actin and myosin. We actually have a fascinating video on that. Transport of oxygen, hemoglobin. So proteins, the way at least my brain thinks of it, they do a lot of the work. The DNA says, well, what contains the information, but a lot of the work of the organism is actually done by the proteins. And as I just said, the building blocks of the proteins are the amino acids. So let's focus on that a little bit. So up here are some examples of amino acids, and there are 20 common amino acids. There are a few more that depending on what organism you look at, and theoretically there could be many more. But in most biological systems, there are 20 common amino acids that the DNA is coding for, and these are 2 of them. So let's just first look at what is common. So we see that both of these, and actually all 3 of this is just a general form. You have an amino group. And this is why we call it an amino acid. So you have an amino group right over here. Now you might say, well, it's called an amino acid. So where is the acid? And that comes from this carboxyl group right over here. So that's why we call it an acid. This carboxyl group is acidic. It likes to donate this proton. And then in between, we have a carbon, and we call that the alpha carbon. And that alpha carbon is bonded. It has a covalent bond to the amino group, a covalent bond to the carboxyl group, and a covalent bond to a hydrogen. Now, from there, that's where you get the variation in the different amino acids. And actually, there's even some exceptions for how the nitrogen is. But for the most part, the variation between the amino acids is what this 4th covalent bond from the alpha carbon does. So you see in serine, you have this, what you could call it an alcohol. You could have an alcohol side chain. In valine right over here, you have a fairly pure hydrocarbon hydrocarbon side chain. And so in general, we refer to these side chains as an R group. And it's these R groups that play a big role in defining the shape of the proteins and how they interact with their environment and the types of things they can do. And you can even see just from these examples how these different side chains might behave differently. This one has an alcohol side chain. And we know that oxygen is electronegative. It likes to hog electrons. It's amazing how much of chemistry or even biology you can deduce from just pure electronegativity. So oxygen likes to hog electrons. You're going to have a partial negative state, a partially negative charge there. Hydrogen likes to hydrogen has a low electronegativity relative to oxygen, so it's going to have its electrons hogged. So you're going to have a partially positive charge, just like that. And so this has a polarity to it, and so it's going to be hydrophilic. It's going to, at least this part of the molecule is going to be able to be attracted and interact with water. And that's in comparison to what we have over here, this hydrocarbon side chain. This has no polarity over here, so this is going to be hydrophobic. So this is going to be hydrophobic. And so when we start talking about the structures of proteins and how the structures of proteins are influenced by its side chains, you can imagine that parts of proteins that have hydrophobic side chains, those are going to want to get onto the inside of the proteins if we're in an aqueous solution, while the ones that are more hydrophilic would want to go onto the outside. And you might have some side chains that are all big and bulky, and so they might make it hard to tightly pack, and then you might have other side chains that are nice and small that make it very easy to pack. So these things really do help define the shape, and we're going to talk about that a lot more when we talk about the structure. But how do these things actually connect? And we're going to go into much more detail in another video. But if you have serine right over here, and then you have valine right over here, they connect through what we call peptide bonds. And a peptide is the term for two or more amino acids connected together. So this would be a dipeptide, and the bond isn't this big. Let me draw it a little bit smaller. So that's serine. This is valine. They can form a peptide bond, and this would be the smallest peptide. This would be a dipeptide right over here. Peptide bond, or sometimes called a peptide linkage. And as this chain forms, that polypeptide, as you add more and more things to it, as you add more amino acids, this can be a protein, or it can be part of a protein that does all of these things. Now, one last thing I want to talk about. This is the way these amino acids have been drawn is the way you'll often see them in a textbook. But at physiological pHs, the pHs inside of your body, which is in that low 7s range, so it's a pH of roughly 7.2 to 7.4. What you have is the carboxyl group right over here is likely to be deprotonated. It's likely to have given away its hydrogen. You're going to find that more likely than when it's going to be in higher concentrations having been deprotonated than being protonated. So at physiological conditions, it's more likely that this oxygen has taken both of those electrons and now has a negative charge. So it's just given away the hydrogen proton, but took that hydrogen's electron. So it might be like this. And then the amino group at physiological pHs, it's likely to actually grab a proton. So nitrogen has an extra lone pair. So it might use that lone pair to grab a proton. In fact, at physiological pHs, you'll find a higher concentration of it having grabbed a proton than not grabbing a proton. So the nitrogen will have grabbed a proton, use its lone pairs to grab a proton, and so it is going to have a positive charge. And so sometimes you will see amino acids described this way. This is actually more accurate for what you're likely to find at physiological conditions. And these molecules have an interesting name. A molecule that is neutral, even though parts of it have charge like this. This is called a zwitterion. That's a fun word. Zwitter ion. And Zwitter in German means hybrid, and ion obviously means that it's going to have charge. And so this has hybrid charge, even though it has charges at these ends. The charges net out to be neutral.